Let us go through muscle physiology real quick. So by the end of this revision, you'll come out very strong, like this guy here. Okay, so what are we going to look at? We're going to go through the introduction, of course, structure of skeletal muscle, the excitation, contraction, coupling of skeletal muscle. Yeah, we have a lot to discuss, but I'll keep it very brief and straight to the point. So to start with, let us have an introduction to muscle physiology. So this is where we just study about muscles in depth. Okay, so we generally have three types of muscles. We have skeletal muscles. So we have skeletal, we have heart muscle. So this is how you spell heart, cardiac. And then we also have smooth muscles. So now what should you know about these kinds of muscles? Yeah, so just generally for skeletal muscles, you have to know that these are voluntary. They are voluntary muscles. So meaning you control how you use your skeletal muscles. Yeah, so these, they are controlled by the somatic motor neurons. Yeah, so in your book you finish that. Somatic motor neurons, they are the ones which control the skeletal muscles during voluntary movement. Yeah, so things like locomotion, when you are moving, respiration, it works under skeletal muscle. And then what else should you know? You should know that skeletal muscles they are striated. Yeah, so this is striated. So skeletal muscles are striated, meaning they have like stripes. I'm going to show you. Yeah, and then what else? Skeletal muscles, they are fast. So these guys are fast and they are strong. Yeah, so this is all you should know about skeletal muscles, generally speaking. And then what about cardiac muscles? So these, they are involuntary. Okay, so by involuntary, you know it means you can't control the heart. You can't say no, because today I'm heartbroken, the heart will stop pumping blood. Yeah, so the muscles in the heart, the cardiac muscles, they are involuntary. Yeah, so in anatomy you are going to learn about the muscles which are found there. Yeah, so these, they are involuntary. You don't control them on your own. Yeah, so no wonder they work even if you're unconscious. Yeah, and then the other thing you should know is that they are striated. Striated. Yeah, just like smooth muscles. So these are striated. Okay, and then what else can I remember? Oh, they are also rhythmic. So the cardiac muscles follow a rhythm. Under we have the use of a stethoscope. We have heart sounds. You are going to learn that on cardiovascular physiology. Yeah, and then now for the smooth muscles, these are also involuntary. So these are involuntary. They are involuntary muscles, meaning you do not control them. And then what you should know about them is that they are slow compared to these others. Smooth muscles are very slow, and they are non-striated, meaning they do not have like stripes. Okay, so these you just have to know. Yeah, these are the basics you should know, generally speaking. And then for smooth muscles, you find them in internal organs like the stomach, the urinary bladder. You have smooth muscles, and then these they are mainly attached to bones. Yeah. Right. So that makes up our introduction. Yeah. So before we go to the structure of the skeletal muscle, I would love to show you just this on the striation of the muscles. Okay. So this is what we've looked at. We've said skeletal muscles, they are striated. Also cardiac muscles, they are striated. But smooth muscles are not striated. Yeah, so you can see cardiac and skeletal, they have stripes. But for the smooth muscles, they do not have stripes. Yeah, and then you can also look at the nucleus. For the skeletal muscle, it is multinucleated. It has a number of nuclei. But for these others, they can just have a single nucleus and all that. Yeah, so that makes up our introduction. So now let's jump to the structure of the skeletal muscle. So this here is our muscle. So we already know this. This is an example of a muscle. So under anatomy, you must have learned on the biceps, the triceps, yeah, all those brachialis muscles. So here we're dealing with physiology. So this is a warm muscle. So when you just take a tiny bit of this muscle, you are going to have what is called a fasciculus. Yeah, so this it is a fasciculus. Yeah, so a fasciculus, so we can say these stripes, like the ones which you can see, those are fasciculi. Yeah, so when we take out one, it is a fasciculus. Yeah, so it is also called fascicles. Okay, so we have a warm muscle. When in a warm muscle, we have different fascicles. This is a fascicle. And then we take it out, that's what we have there, a fasciculus. So now this is under magnification. Okay, so we have a fasciculus, and then inside of the fasciculus, you are going to find now the muscle fibers. Yeah, so these are the muscle fibers. So now from the muscle fibers, we have a myofibril. Yeah, so we now take another branch. Okay, so and then now from the same myofibril, we have what is called a sarcomere. So a sarcomere, it is a functional unit of a muscle. Remember, like if we're talking about a cell, the functional unit of living organisms. So now here we have a sarcomere. This is the functional unit of a muscle. Yeah, you can write down that. So this is how a sarcomere will look. It will look like this. It has dark bands and light bands. So now when we magnify further into the sarcomere, we're going to see myofilaments. Yeah, so now the myofilaments are what we're now going to discuss in more depth now. So let us talk about the myofilaments. So myofilaments, we're going to make this real quick. So myofilaments, we have two. We have what are called thick myofilaments. So the thick myofilaments, they are made up of myosin. They are found in the thick filaments. Yeah, and then the other type of myofilament, we have the thin. The thin filament, it has three things. We have actin, and then we have tropomyosin. So you can see tropomyosin, meaning it will relate somehow to the myosin. And then the third one, we have the troponin. troponin complex. Okay, so this is what we find. So this is a myofilament. Yeah, I'm going to show you better pictures. So that's a myofilament. It is made up of two types. We have the thick filament, thin filament. Thick only has myosin. Thin, it has these three things. Are we good to go? If you've understood up to that part, you can like the video. So this is the this is now the general summary of the structure of a sarcomere. So of the sarcomere, here I don't want to focus much on the light band. Yeah, for our revision's sake, you can read further on that. So we're interested in the myofilament. We have thick and thin. We said for the thick, it has myosin. So this is myosin. And then I've even remembered that I forgot to say one thing. So for the myosin, so on the myosin, a myosin has two things. It has a head, bilateral head, and a tail. Yeah, so the heads are two. So this is a myosin. Yeah, so a myosin has two heads and a tail. Okay, and then for the thin filament, we already said we have troponin, actin, tropomyosin. Yeah, so what are these same things? So for the we can start with which one? The actin. The actin are these the actin are these green parts you can see. And then the troponin, it is right here. Those are troponin complexes. And then tropomyosin, it is the orange line. So this orange line you can see, that's the tropomyosin. Okay, so that makes up our sarcomere. Okay, so we have discussed in detail the structure of skeletal muscle. So this means that you are now ready for the excitation, contraction, coupling. So here I need your ears. This is a very important part. Yeah, so a question, maybe they'll give you a kind of scenario where they want you to explain how a muscle contracts. So lecturers are not kind people. They cannot ask you a question like they can't ask you something like how does a muscle contract. No, they can't ask you like that. They'll give you maybe a scenario and then they tell you to explain the mechanism. Yeah, unless if they are kind, they tell you exactly that. Explain the excitation, contraction. Yeah, so this is just how a muscle contracts. Yeah, so now let's go through it step by step and real quick. So the contraction of a muscle, the story starts all the way from a neuromuscular junction. Yeah, so here now we're going to bring back the knowledge of the action potentials. Yeah, so we're going to have a neuromuscular junction like that. Okay, so in your book you're going to draw something better. Yeah, so in the exam or in your test, it can even be a good thing if you draw. Yeah, so this, it is a muscle fiber. Okay, so now how does the story play? So the story starts with an action potential. I'm just going to write AP. So an action potential comes, it travels along the axon terminal. So now when the action potential reaches here, it is going to stimulate the release of a neurotransmitter known as acetylcholine. Yeah, so acetylcholine is going to be released. So now acetylcholine, and then when it passes, it comes to the other side. It is going to bind to nicotinic. It is going to bind to nicotinic receptors. So now this is called the postsynaptic cleft. Yeah, it's going to bind to nicotinic receptors. So now what happens when acetylcholine binds to the nicotinic receptors? It is now going to lead to the formation of an end platelet potential. Yeah, so we have an end platelet potential. It is like an action potential, but it is on the motor end plate. Yeah, so the end platelet potential, it is now going to travel along the muscle fiber. So this will result in depolarization of the membrane. So when there is depolarization of the membrane, we are going to have a situation where calcium or potassium will be leaking out, while sodium it is going in like that. Yeah, so you have to be explaining this. You can be writing in point form or paragraphs. Yeah, so this will now lead to a depolarization of this whole thing. So now the action potential will travel along the T, it is called the T-tubule. So this thing, it is a T-tubule because it looks like T. It is like letter T. So this is the T-tubule. The action potential will travel down. So now it is going to find a gate here. It is called DHP. This is Dihydropyridine Receptor. Okay, so and then I even forgot a structure. So we have a structure. This is called the Sarcoplasmic Reticulum. Is it Sarcoplasmic Reticulum? Okay, so action potentials travel down here, and then there's going to be, they are going to open the voltage gated ion channels. Yeah, so now this it will lead to the opening of another gate here. The gate is called RYR, which is Ryanodine Receptors. Yeah, so you can check the description of this video. I've put the full meaning of these terms. Okay, so when Ryanodine receptor, when it is opened, so in here in the sarcoplasmic reticulum we have calcium. So calcium is now going to escape. You see that? So calcium will escape the sarcoplasmic reticulum. It goes to the outside world. So now when calcium goes out, we're now going to have a different party. So now the muscle is now getting excited. Remember the heading where we are, excitation. Yeah, so this happens now when calcium leaks out, meaning the party is about to officially start. So when calcium flows out, it is going to bind to troponin. Troponin C. So remember we talked about the thin filament. The thin filament has actin. What else? It has actin, tropomyosin, and troponin. So calcium, it is going to bind to troponin C. And then now when it binds to, let me show you troponin C again. So this here is the what we looked at. This is the thin filament. So the thin filament has troponin. So troponin actually has three parts. It has troponin I, troponin is it troponin U? I've forgotten the troponins. What is yeah, but I can just remember of troponin I, troponin C. I think the other one may be troponin U. Yeah, so I'm not going to discuss this in detail. So here, just know that on the troponin, there are three parts. So calcium, it is going to bind to troponin C. Yeah, so in case you find this as a multiple choice question. Okay, so we're saying, so just a quick recap. When the action potentials reach this gate, dihydropyridine receptor, when it opens, it is going to stimulate the mechanical opening of the ryanodine receptor. So when the ryanodine receptor is opened, calcium will leak out. When calcium leaks out, it is going to bind to troponin C. This is where we are. So now what happens when calcium binds to troponin C? We're going to have, you can see this tropomyosin. Tropomyosin, I said it is this orange line. It is going to move out from the actin. We said actin, it is this green bubble. Yeah, so meaning at first, the way these things look, they look like this. But when calcium binds to troponin C, the tropomyosin is going to move away from the opening, meaning the actin will now remain opened. They are not closed, like they are not covered. Yeah, so now what happens when the myosin is not covered? This is what, it's actin rather. Actin is not covered. When actin is not covered, or the myosin, it will come and bind. Okay, so myosin, remember, it has two heads and a tail. So myosin will bind. Yeah, and then when myosin binds, it will produce a power stroke, whereby the sarcomere is going to reduce. Like there's going to be pulling. The myosin will pull the sarcomere. Yeah, it will reduce in strength. So that is a power stroke, meaning a muscle has contracted. Yeah, and then next there'll be cross bridging, where there'll be later on there'll be release of the myosin and all that. Yeah, so this is how a muscle contracts. A question, usually they don't say that, how does a muscle contract? You can be asked to explain the excitation, contraction, coupling. Yeah, so that's a summary of what you should know about that. Yeah, so remember, we're just having a very rapid revision. You can check out other videos later on where we discuss in more detail. So now we are done with this, no wasting time. And then mechanisms of muscle contraction. Okay, so this one I'm going to make it real quick. So what are the mechanisms of muscle contraction? So let me just rub this. So on the mechanism of muscle contraction, we're just going to quickly go through my old physiology book. Yeah, so this is a summary. We have, I don't know how clear it is. We have cross bridge formation. Then there will be a power stroke. Then there will be cross bridge detachment, and then there will be reactivation of myosin heads. Yeah, so now just to quickly explain, in case you are asked something like this. So for the cross bridge formation, this is just where can I write? Okay. The cross bridge formation, this is where we have calcium. Remember the whole thing, when calcium is released from the sarcoplasmic reticulum, we said it binds to troponin C. When it binds to troponin C, there's going to be tropomyosin movement, meaning tropomyosin will move away from the actin. Yeah, so now when tropomyosin moves away, we're going to have the myosin head binding. Yeah, so I'm sure you have gotten the picture. These things, they look like this. This is actin, this is tropomyosin. When calcium binds, the tropomyosin goes away. Yeah, And then now when the tropomyosin goes away, it means the head of the myosin are going to attach. So now when the heads attach, we are now going to have a power stroke. Power stroke is whereby these they'll be moved. Yeah. Myosin will be released, and this happens, it releases rather ADP. Yeah, and then that's when the actin is going to be pulled towards the center of the sarcomere. Okay, so that's a power stroke. Power stroke simply means that the actin is pushed. Okay, so now what happens after the power stroke? We're going to have so now let's go through the cross bridge detachment. So here is whereby the ATP, where can I show this? Okay, so let's do this. Okay, so here I want to make this clear so that you follow it well. So we've talked about the cross bridge formation. This is where myosin binds to actin. Actin are these round things, and then when it binds, it produces a power stroke, meaning these they'll be pushed. Yeah, so like let's say even here, just the way it is, these three they'll be pushed, meaning they'll come somewhere here, meaning it's a power stroke. Yeah. So now what happens next? So after the power stroke, let us now talk about the cross bridge detachment. Okay, so this remember, this is the actin, the one in green, the one in orange, that is the tropomyosin. So now the stage where we are, so here this is where we talked about the power stroke. So the power stroke meaning this whole thing, it has been moved, especially to the left hand side. That's a power stroke. So now what happens next in the mechanism of this contraction? We're now going to have a cross bridge formation, cross bridge detachment. So this is myosin. So what happens? For myosin to be released, there's going to be binding of ATP. So ATP, we can just say this is, and for example, let's say we have this guy here with this girl, and then the two, they are kissing. Now this girl, she has a younger sister or younger brother. When the younger brother comes, obviously the two people are kissing, they'll stop. That's how ATP acts. So think of ATP as the little sister. When it sees the two kissing, they'll stop. So when ATP binds to myosin, the heads will let go. Like, okay, we can't kiss anymore. Yeah, that's what happens. And this is called cross bridge detachment, because the heads are being detached from the actin. Yeah, and then the last step, reactivation of the myosin head. This is where ATP is hydrolyzed and it's going to be ready for the next cycle. So this is how a muscle contracts. So now to put it in simple terms, the way a muscle contracts, I wish we could make it here, but it would take time. I would generate a video where it's showing how this happens. So, but just for simplicity, we're going to do this. So now the way a muscle contracts, it is when myosin is going to push the actin to the other side. Yeah, because when it pushes the myosin, or when it pushes the actin, it means the sarcomere is going to reduce in length, meaning a muscle will contract. So that's how a muscle contracts. Yeah, so I think we have provided much detail, coming all the way from calcium, troponin C, troponin, tropomyosin, all those things. Okay, so now with that, we can go to the next item. Yeah, so we've handled mechanisms, neuromuscular diseases. But before the neuromuscular diseases, I would like to quickly talk about the length-tension relationship. So we come back to my book. Sorry, my handwriting was very bad. I was practicing to be a doctor. Yeah, so quickly going through the types of muscle contraction. We have two types. We have isometric and isotonic contraction. So isotonic, so whenever you see isotonic solution, it simply means that there's something which is constant, meaning it's just the same the way it is. So now in relation to tension, isotonic means the tension will be the same. Okay, and then our metric, we now have a variation. So isometric contractions, this is where tension develops within the muscle. Okay, so let's take this hand. Okay, we'll start with isometric. So this is where we have tension. Yes, so under isometric we have tension, it's there, but length remains constant. Yeah, so this is an isometric contraction. So how can we explain this? We can say, for example, we have this little guy, Ella. Oh, yeah, sorry for that guy. He's making noise. Me, I'm trying to have fun revising. Okay, so quickly, we have this guy here. Let's say he has been punished to carry a big block. So now what you expect, yeah, excuse me. So what you expect here is that since he's carrying a heavy load, there's going to be tension, but the muscles, they are not going to be lengthened. This is isometric. Yeah, so you can even be given such a scenario. You have to understand tension will increase, but length will not increase. Quickly going to the other one, isotonic. Isotonic, always remember that tension remains constant. It does not increase, but the length of the muscle will increase. For example, if this same person starts putting down the block, meaning the hands, they are stretching. So when the strength is lengthening, we have isotonic. So the strength is lengthening, but the tension will not match. The tension will not match increase. Yeah. So for here, in such an example where I said the hands are lengthening, when the hands are lengthening but tension remains the same, or it's developing, tension is developing, but the length of the muscle is increasing, we call it eccentric contraction. Yeah. So and then the other type of isotonic, we have concentric. So this is eccentric, this is concentric. So the tension develops in the muscle, but how the length is. So here the whole thing, you just have to know on the length. Yeah, so the length, when the length doesn't change, we call it isometric. But when the length changes, it is isotonic. And of course, tension would remain constant. So here I wrote tension develops, meaning if tension is constant, it doesn't mean that there's no tension. Yeah, so it is there, it develops, but it will remain constant. Yeah, so now how does the length increase? If it shortens, it is concentric. Lengthens, it is eccentric. Yeah, that's the whole essence. So just a quick summary. If the length, so when we're talking about the length, so if the length remains constant, like you are lifting something, if the length remains constant, that is isometric. but if the length changes, it is isotonic. Now depending on the change, if it shortens, if we say the length becomes short, it is concentric, and then if the length becomes long, it is eccentric. Okay, so with that, I think we can now quickly go to the next part. Here now, the neuromuscular diseases, you have to pay particular attention to this part. So with that, let's go to the first one already. So the first neuromuscular disease we're going to discuss my asthenia gravis. So we have my asthenia gravis. So this one, it is one of the most important. Yeah, together with the second one which we'll talk about. Yeah, so ensure you will write down what I'll explain on this one. So my asthenia gravis, first thing to take note of here is that it is an autoimmune disease. So meaning it is as a result of the immune system reacting on its own. Yeah, so asthenia gravis, it is an autoimmune disease of the neuromuscular junction. So do you remember the neuromuscular junction where we have this? Yeah, so this is the neuromuscular junction. Yeah, so it is an autoimmune disease about this. So now it is whereby the cause of asthenia gravis. So the cause of asthenia gravis, it is development of antibodies, especially the IgG. Yeah, so when you do blood physiology and also when you do immunology in third year, you are going to learn about the different immunoglobulins. Yeah, so here just for our summary, just know that when there is development of immunoglobulin, how do you write it? Immunoglobulin G. Yeah, so I'm going to discuss this in more detail ahead. Yeah, so when there is development of immunoglobulin G, then when the antibodies are produced, it can lead to asthenia gravis. Yeah, so now how it comes about, it is when there's prevention of binding of acetylcholine to the receptors. Yeah, so when you're explaining, ensure you mention this. This is very important, even if you even forget this. So when there's prevention of acetylcholine, when it doesn't bind, remember it has to bind to the nicotinic receptors, but when it doesn't bind, then we are going to have now asthenia gravis, which will come into effect because of the non-binding. Yeah. So I saw a question in some past paper where they just gave symptoms. Yeah, so I think I sent the paper in the group. If you're not in the group, you can let me know and then I'll add you there. So you have to know the signs and symptoms, because clinical scenario questions, they'll give you symptoms and then they tell you what is the likely diagnosis. Okay, so let's quickly go through the symptoms of asthenia gravis. We have slow muscle contraction. We have weak muscle contraction. You find that someone's muscles, they are quite weak. They do not contract to their intended capacity, if I can say. Yeah, so there's slow muscle contraction, weak muscle contraction, and then there's inability to control prolonged contraction. So you can take, for example, we have this guy. When we give him a box, maybe the box is not even so heavy, but when this person lifts the box for only like 20 minutes, they are getting tired. So there's inability. That's the third symptom, inability to control prolonged contraction. They are not able to control having their muscles contracted for a long period. Yeah, so ensure you are writing this in your book. And then we also have quick fragility. Quick. How do you write quick? So we have quick fragility. So quick fragility, fragile, whereby we can say you are getting tired so fast, in simple terms. And then there's also difficulty in speech. Yeah, so these are just some other signs and symptoms. Okay, so you have to make sure that you know those. And then now you will also be asked about the treatment. Okay, they give you the signs. From the signs you have identified that, okay, it's myasthenia gravis. Okay, so how can you treat it? Myasthenia gravis, it is treated by substances such as neostigmine. So this is a drug, neostigmine. And then there's also so this is pyridostigmine. Okay, so I know you do a good job in writing neatly in your books. So here, how myasthenia gravis can be treated is through the one of the ways it is by administering these drugs. So now, how do these drugs even work? So firstly, you have to understand how myasthenia gravis is caused. Remember, I only said that acetylcholine is not able to bind to the nicotinic receptors. So now, do you know what causes that? There's an enzyme called Acetylcholinesterase. Yeah, so the enzyme, it inhibits acetylcholine from binding. So now, with these drugs, they're going to help the acetylcholine to bind more effectively. And they are going to ensure that more acetylcholine is produced or more acetylcholine is released, so that at least the chances of binding are increased, meaning even at least the muscle contraction it will be there to some effect. Yeah, so you can be asked what drugs can be administered. Yeah, so if you go through some of the past papers, there are questions about this. Yeah, so that's a way in which it can be treated. Okay, so now we quickly rush to other neuromuscular diseases. So the second one, it is also an important one. It is also the one which we'll discuss in detail. Yeah, for the others, I'll just list them and then maybe just a brief talk on them. So we have what is called Lambert-Eaton, myasthenic, yeah, from myasthenia gravis, like myasthenic syndrome. So this one in my book, I used to write it as LEMS, Lambert-Eaton myasthenic syndrome. Yeah, so these are abbreviations I'm going to to put them in the description. Yeah, so this is a disorder of the neuromuscular junction, which is caused by development of antibodies against calcium channels. Yeah, so it is antibodies are developed against calcium channels. Remember, these diseases are autoimmune. So when we said autoimmune diseases, it means antibodies are at work. Your own immune system are causing you to suffer, if we can say it like that. Yeah, so now when the antibodies are produced against the calcium, and remember, calcium, it has to bind to troponin C, and when calcium binds to troponin C, we said tropomyosin is going to leave the actin. Yeah, I hope you remember that. Tropomyosin, so ensure you study. Tropomyosin will leave the actin site. The actin site, when it is free, it means the myosin heads, they are going to bind. The myosin heads are going to bind, meaning we are going to have a power stroke. So now, without calcium, meaning we are not going to have a power stroke. Without a power stroke, there's not going to be contraction. So this is how Lambert-Eaton myasthenic syndrome is all about. Calcium is inhibited. Okay, so we can even check the exam paper for 2024. Things on this were part of section B. Yeah, so we'll talk about that later. Let's quickly conclude. And then the symptoms. You always have to know the symptoms. They can give you symptoms for Lambert-Eaton, and then you say it's myasthenia. So the symptoms, they are just very similar to myasthenia gravis. Actually, all those symptoms we said slow muscle contraction, weak muscle contraction, quick fragility, someone is getting tired fast, not able to control long contraction. All those, they are also symptoms here. But in addition to all those, we have blurred vision. Someone is not able to see properly, and also dry mouth. So if they give you all those symptoms for myasthenia gravis, then you just know it's myasthenia gravis. But if they just include some of these, you have to say it is Lambert-Eaton's myasthenic syndrome. Yeah, and then sometimes they can give you, okay, yeah. So I'm now guiding you on how tricky sometimes the exam can be. They can give you symptoms. They give you symptoms which are, let's say, they just give you 2 symptoms. Yeah, so as trained, let's say they tell you, let's also rub blurry vision. Yeah, so but just know blurry vision and dry mouth, they are Lambert. So what can you do if they tell you the patient has slow muscle contraction, weak muscle contraction, and also weak arms? Yeah, weak arms and weak legs, they are symptoms. Yeah, they give you all these, and someone cannot stay for long. So now, can you say it is myasthenia gravis, or you say it is Lambert-Eaton myasthenic syndrome? So if the symptoms are like this, then you can just pick any. But if they just say blurry vision, just know it is Lambert. Or if they say dry mouth, just know it is Lambert. And then if they say maybe they have given you these symptoms, and then they tell you that acetylcholine is not able to bind, you have to know, okay, if acetylcholine is not binding, then if acetylcholine is not binding, then it is myasthenia gravis. And then if calcium is not binding to troponin C, it is Lambert-Eaton. Yeah, I think I'm very clear there. If you've understood this part, don't forget to like the video. And then I will quickly go to the other side. Some other diseases, we have neonatal, what is it called? Neonatal myasthenia gravis. Yeah, it is just like myasthenia gravis, but when you see neonatal, this is children. So this is like myasthenia gravis in children, in babies. So here I'm not going to write just a quick summary. Yeah. So it happens in newborn. So when a mother, let's say a mother has myasthenia gravis, then the child now will develop it. Remember, since this is from the immune system, so it can affect the child. So when a mother with myasthenia gravis gives birth, the child will have neonatal myasthenia gravis. Yeah, and the signs and symptoms of neonatal myasthenia gravis is whereby the child has a weak cry. They are not screaming so much. The crying is very weak. Yeah, and when there's also poor sucking, just from the way the child is sucking, if it is not, they just have poor sucking. Yeah, it is a sign of neonatal myasthenia gravis. This is G. Yeah. So we can go to the others. So remember, these are minor neuromuscular diseases. So I didn't plan on spending more time. So now let's go to what is called neuromyotona. Yeah, so these terms, they give you reason why pharmacists and doctors, they have bad handwritings. So neuro, they're supposed to be O. Neuromyotona, or just neuromyotona, it is also called Dissec's syndrome. So now when you just see neuro, just know it is dealing with the brain, or mainly the nerves, the central nervous system. So what is neuromyotona syndrome? So this is where we just have hyperexcitability. So meaning, remember the excitation coupling system, but now if it is very high, like the contraction are very much high, it is neuromyotona. It is because of a peripheral nerve which is disordered, and then it is resulting in hyperexcitability. The action potentials are way too much. Yeah, neuromyotona. So the causes, it is antibodies against calcium channels. Yeah, and then the signs and symptoms. Signs are muscle cramps. Yeah, if there are muscle cramps, it is neuromyotona. Yeah, and then to quickly finish it up now, we have what is called the Duchenne muscular syndrome. Syndrome, you finish on your own. Yeah, so what is Duchenne muscular syndrome? Oh, and I was busy saying syndrome. Yeah, so it's Duchenne muscular dystrophy. Yeah, so I don't know if you know the spelling of dystrophy, but I know you know. Yeah, so what it is abbreviated as DMD, Duchenne muscular dystrophy. So here, this one, it is not an autoimmune, meaning antibodies are not involved like all these others. So this one, Duchenne muscular dystrophy, it is whereby we have X-linked recessive, like you remember from genetics. Yeah, these things. So it is where now there's a mutation in dystrophin. Yeah, and then now for the signs and symptoms, there's what they call Goer's sign. So if you just see Goer's sign, the disease is this one. So Goer's sign, it is just whereby a child, oh, by the way, this one, it is common in children from 2 years to 5 years. So Duchenne muscular dystrophy is whereby a child is not able to stand on their own. Maybe if they are trying to stand, they grab a chair, they grab something so that they can stand. someone is 5 years and then they are not able to stand properly, they have to get support. That is Goer's sign. Yeah. And then some other symptoms, there can be prolonged weakness. Yeah, so these are the neuromuscular diseases. For our revision, we'll end there. Yeah. So you can read further. And then types of muscle contraction. I think we talked about them already. Isometric, isotonic. Yeah, those. And then cardiac and smooth muscle. So this one I'll be very rapid. So now, cardiac muscles. The most important thing here is for you to understand the electrical property of the heart. Yeah, so but before that, we can even quickly talk about the types of muscle fibers. So the types of muscle fibers, we have what we call the contractile unit. Contractile unit of the heart. It is a kind of muscle fiber. And then there's also the pacemaker. So you know the A node, AV node. Yeah, those we're going to discuss them in CVS, the cardiovascular system. Yeah, so but just listen, we have the pacemaker, we have what we call pacemaker cells. Yeah, so these are a kind of fibers. And then the other type of fiber, it is the conducting system. Okay, so this is not a main focus for this lesson. I just thought of mentioning it. So what I want you to understand very well here is the electrical activity of the heart. So we have phases. We have phase 0, we have phase 1, and then we have phase 2, we have phase 3. And then we also have phase. So phase 3 it is here. And then we have phase 4. So what I'm going to explain, quickly just write it for your own revision. So starting with phase 0, this is where the first gated sodium channels are opened. When the first gated potassium channels are opened, there's going to be influx of sodium, which means it is going to increase the resting membrane potential. The resting membrane potential will reach somewhere up to between 20 and 30 millivolts. Remember, the normal resting membrane potential of cardiac muscles is negative 90 millivolts. Yeah, so but when sodium is when the gates are opened, then the resting membrane potential will be increased away from its normal, which will take us to phase 1. Phase 1, it's called initial polarization. So you just hear initial polarization, meaning, this is repolarization. Initial repolarization. So just know that there's going to be another repolarization. So initial repolarization, it is whereby we have sodium will be reduced, but potassium is increased. So I'm trying to write potassium. Potassium is increased. Remember, because the channels are opened under potassium, it is rapid repolarization. This potassium increase. And then phase 2, it is called the plateau phase, because it is almost like constant. Here there's increase in calcium and decrease in sodium, meaning the gates for sodium are now closing. Yeah, and then phase 3, rapid repolarization. Calcium will decrease further, calcium will increase further, and then potassium will decrease further. And then the last phase, which is phase 0, it is whereby the resting membrane potential tends to 90. Okay, and then from there we can quickly go to the smooth muscle. Okay, here now I'm quite tired already. Yeah, so just pause the video, screenshot, you read through this. When you have questions, you let me know. So this is the mechanism of muscle contraction, how it contracts, how it relaxes. And then the sources of energy were phosphocreatine, glycogen, and aerobic system.